So, hi everybody. This is Ricardo Borges. This is a presentation for pyMETAflow for NMR, which is a pipeline I created to treat NMR traces for metabolomics and then data comparison. Really, this is a version for NMR. I have a version for HPLC traces as well, and this can be really updated for any kind of trace, for different samples for metabolomics type of work. So the goal here is to transform processed NMR data into useful information for sample composition and biological variations. It's meant to be a complete workflow for NMR data analysis and comparison, right? But it still needs the NMR data to be properly processed beforehand, like using MNova, for example, right? Well, in Brazil, we are used to use MNova for NMR data processing, and this was my starting point. So we're gonna see that we are using as an input the CSV file exported from MNova. So about the main features, we have data imports and post-processing part. I'm saying post-processing just because you firstly do the MNova processing part, and then this part, and then the data analysis. So we do importing plots for NMR spectra, filter chemical shift range, and remove noise or any unwanted regions. We allow for exclusion of specific samples from the data set. It's always better to do that programmatically instead of just erasing data. Right, and it applies data transformations and normalization and scaling before the statistical analysis. For the data analysis, we support multivariate analysis such as PCA, PLS-DA, and some more, and we are always increasing the range for that. We include data fusion tools, enabling the integration with different information source like biological activity for natural products, or chemical profiles, for example, or HPLC traces, or even LCMS. And we generate plots and data visualization to facilitate the results interpretation. This is always nice, and everything gets documented. So as a requirement, we need the data to be processed, and for this part, we need it to be processed on MNova just because of the format. And so you would make FT processing, phase correction, referencing, baseline correction, whatever you like. And then you would select the superimpose mode for the samples, the data you need, and you export the superimposed data as a NMR CSV file, right? So this data will be the input data that we need. Another requirement that we have is that you need to install Python, and we suggest that we're just saying that this was tested on the Python 3.11.6. Right, here is the link for you to download. And concerning the organization setup, you would download the pyMETAflow zip files from GitHub. Here's the link, and you will organize it in that manner. You can put the pyMETAflow folder in any place you'd like, and you should have these data processing NMR.py for the Python functions, a README file, the requirements.txt. This is the Python packages dependencies just for the installation, and the run.bat, that would be for Windows, and you would run that uh, to install everything, even the Jupyter notebook. Once it once everything is installed, when you double click this run.bat, it would recognize that the environment is already created, and it will open the Jupyter notebook, right? So your data, the user data, would be placed on specific project folders, and here an example for that would be the project folder in here, and we should have each process. Each project should have the same its own Jupyter Notebook file to avoid any confusion. The user should have a metadata.csv file. Well, this is good for any study, right? But you would need samples, LC file name, NMR file name, or attribute for classification. That will be somewhat required. The samples and the file name is requirements, and the attribute classification for the classes, right? And well, of course, the NMR data as exported from MNova. Of course, we can create variations for the import functions to get data from different sources as well. But that will be in the future, right? So that would be the GitHub web page, right? You would click on the code, this green button here. It's a drop down button, and then you would select download zip, right? This will download your zip folder, and everything will be inside. That is really the simple thing to do. You can import that as a GitHub desktop thing, and but only if you really want to change something and suggest me some changes, which are always welcome. So the starting point is the importing of the NMR data and plotting spectra. So we have a function for import that imports a CSV file, the file exported from MNova. We can apply chemical shift filters by removing ends and specific regions on the spectra. Things like solvent, the peak of the solvents, or water from the solvent, or even the DSS. But I wouldn't remove the DSS just now, just because we can use it. We have to use it for referencing, and so well, each case will have its own specificities. We have another function to exclude samples, like you have blank samples or quality control samples that sometimes you want to remove from your data set, just because you already check it that the blank sample is blank and the quality control samples are are good enough. So you want to um, exclude them for you to be able to go through your analysis, right? And we make plots, some decent plots. All the plots are saved as HTML, so they are high resolution, and you can zoom and check for chemical shifts, and you can remove samples from the plot itself, or adding samples from the plot. You can do all sort of things, and then you can download the figure, the image as a PNG, right, um, for publication or documentation. So this is an example for the plot that we get. Later I'll show demonstration, and I can zoom in and let you know show you guys how to deal with it and how to download it. It's fairly easy. So we can also correct chemical shift variation across executions, across runs, and that will be the data referencing. This is different from an alignment, but this is just referencing. you can do that fairly easy on the NMR data processing tool like MNova, but I would just do it again here. So that function will select any peak that occurs between a certain x limit. So you can see minus 0.2 to plus 0.2. That would be the region for where we are. It would find the peak, and that will set then it will set that peak for 0.0, right, as DSS should be. So now that the data is referenced, the useless parts are removed and the samples are filtered, the useless samples, you can just go for the alignment, right? And that was a big part of this because alignment is not very well done by MNova. So okay, we created this function that includes different methods, mainly IcoShift, Raft, and Paft. They are available for the alignment. And then we create a multiplot for you to decide which alignment you want, right? And which alignment better fit your data, and then you go from there. So just to be more specific, the Raft method is the recursive alignment by fast Fourier transform. IcoShift is the interval correlation optimized shifting, and PAFT is a piecewise alignment by fast Fourier transform. So what we need to know is that Raft is a little bit more computational heavily, and PAF may lose global alignment context, but that's faster than Haft. And IcoShift is simple. It's more simple, right? It takes a moment just because of the warning plots, and may not perform that well for heavily overlapped signals. Mostly I've been using PAFT, right? And then you move on for data transformation. For normalization, we have z-score normalization, PQN, and some control-based normalization. This is used to correct systematic variation in the data, such as injection volume, sample concentration, or detector intensity, these kind of things, making the samples more comparable between each other, right? Just before the statistical analysis. Actually, we do that before scaling, but I don't think it does matter which one you do first. So z-score would subtract the mean and divided by the standard deviation, right? Per variable. Um, PQN adjusts each sample by a scaling factor based on its typical profile that would be calculated by the median of quotients relative to a reference sample. That is quite robust. I'm finding really useful for NMR. Control-based normalization uses one or more samples as a reference, right? And that's easier to interpret. Um, And once you choose the method that fits your data the best for normalization and scaling, you can just plot the before and after. That would be quite similar to meta-analysis normalization scheme, right? You can plot the data and check for the normal distribution of your samples and variables, right? And these image is saved. One thing that I forgot to mention is that every image is saved on a subfolder, subdirectory, named images, and each data that is created is saved on the data directory. So one thing that I need to mention is that we created a function to format your data according to the meta-analysis format, right? For you to be able to export your data from here into meta-analysis for analysis. So if you don't if you don't trust these things, well, I did validate it, but if you don't if you want to do your stuff on meta-analysis, you can go ahead and do it there. But I would say that after the analysis, the alignment is really key for full resolution analysis, and MNova doesn't do that pretty well. So that's why people are used to being their data. So once your data is not very well aligned, you would go for binning, right? But if it's the alignment works nicely, you can go for full resolution, and the results are really much better. Um, so you can feel free to use pyMETAflow just for the alignment case, but we'll go ahead and go for the data analysis from here, because you can do easily PCA and PLS-DA from here directly. So to add a step for quality control, we have this quality control check that we added. Here we would filter all the samples according to the classification, and in our case, we have the QC pool sample. They are classified as total pool, right? And these samples would represent the whole study, right? So we get these spectra, this data set, and plot the distribution of each peak, right? We can plot the spectra that we save it as QC spectra that you would check for overlapping. You would imagine that every spectra from the QC would be the same, or you can select regions for specific peaks of interest and check for the distribution of their area under the curve for each sample, and check for the standard deviation. You can see that a standard deviation as low as that, it's a pretty good quality control. So for the data analysis, we have PCA, PLS-DA, Stoxy, and OPLS-DA in our pipeline. I won't go dive into each method, but yeah, well, this is a demonstration of how the PCA score plot looks like. It is classified. The color code is defined by the classification column from the metadata, and we can plot the loadings as a spectra. That is pretty useful for us to check which peak is actually being responsible for the grouping or the differentiation, right? Just an example, if you run that on your meta-analysis, you will have like a value for a beam, and that is not very useful. It's always better to check NMR spectra as a spectra, right? Just remember, you all, PCA is unsupervised, so it won't use the classification information. It just use the data itself. So we are using the classification just for the color coding. For the PLS-DA, now we are talking about a supervised analysis. We actually use the classification vector, that would be the classification column from the metadata, in the calculations. And so you need to have that settled, and we make up an additional plot, um, not only the loading plot but the validation plot for the Q and R square, right? Because you need to check the validation to avoid overfitting, not to avoid overfitting, because you don't avoid overfitting by checking the value, but you verify um, for overfit in your data. But in any case, it's always useful to check the PCA before the PLS-DA, right? Because if the PCA is good, the PLS-DA is probably good as well. And if the PCA is bad, the PLS-DA is probably overfitted. So one function that I like a lot for NMR is the Stoxy function, statistical total correlation spectroscopy, that calculates the correlation of each variable from one variable chosen as a target, right? So the plot is, it looks like that, is modulated. It has its intensity modulated by the covariance value, mostly positive versus negative, and it's color coded by the correlation value from the driver, right, from the target peak of the selected chemical shift. That is quite useful to connect different peaks that might characterize a unique compound, maybe a driver, or maybe a biomarker, or something that interests the user. And as an additional part, and that would be something, some differential for this pipeline versus the others, and that would certainly not be doable on MetaboAnalyst. It's the possibility for data fusion. So um, implementing the data discovery, the depth discovery pipeline, we can integrate chemical profiles from NMR with bioactivity. That would be mainly for natural product researchers. And before metabolomics, we could integrate chemical profile, well, actually the NMR data, with some phenotypical data, right? That would reveal chemical correlations between compounds and between uh, or the bioactivity to the chemical profile, or the phenotypic data with the profile. So for that, we absolutely need the metadata, just to organize the samples and the data from each sample, and we need an additional data for the bioactivity data or phenotypical data. So that would be another CSV file that will look like that, and that table, there is a vector actually, uh, where each column has a sample name, a sample code, and that code would be included in the metadata as a column for bioactive file name. So your metadata would have some sample specification, some ID that won't be used, some attribute clustering um, vector, that would be the classification vector, the NMR file name to put everything in order according to that would be the same file name exported from MNova on their CSV file, and the bioactive file name. And then you can choose to do the Stoxy from the bioactivity uh, data. Well, that would be a vector of variable numbers, right? and then your stoxy would become shy, like statistical retrospectroscopy, and then you would get the highly correlated and covariate peaks or profile from the NMR data to your activity. That would be useful, for example, for um, for you to check which compounds can be associated to a bioactivity or a phenotypic evaluation. And you can actually add another spectroscopy technique from that. You can actually add LCMS features into your analysis and calculate the correlations from your LCMS data to your NMR data to improve your identification routine or confidence, right? And once again, you can export your data in a metable analysis format. That you can do whatever other stuff that you want. It's, it's just on metabolism format because I like that format. It's actually a general data format that has the classification for each sample, right? So this is it. In the next slides, I'll be doing some demonstration with the pipeline itself. So let's make it a demonstration, right? We have the pyMETAflow downloaded here and placing it on well, my documents, right? I'll double click run. This is a Windows. I'll double click run and it will, well, run the script, a script that will install everything that we need. We only need Python, and I put the version there. But yeah, after you install everything, that run program will just add open the Jupyter notebook, right? So we go for our project. In this case, this is the project. These are downloaded these data from the metabolomics workbench. This is it. Okay, yeah, this is it. Smoking and urinary metabolomics. But I filtered just for the smoking NMR data, right? So let's leave that aside. And okay, this is the folder, the main folder that we have. So we have metadata. Let me show you the metadata, how it looks like. Um, just a moment, just for me to make it right, for you to visualize it. Semicolon. Okay. So this is the metadata. You have some sample specification, some ID, attribute group. This is for classification, right? For color coding the PCA and for the PLS-DA. This is the NMR file, the file name, and this is the bioactive file name. This is just a mock data that I added here just to provide you with the final part, right? And the bioactive data, that's a CSV file that looks like that. Just a mock data. For each sample, we have a number, and these sample names, that will be the bioactive file name. Okay. Okay, let's move forward. So we have the CSV file with the NMR data itself. That's the MNova file. I won't show you that just because we don't need to see that right now. So when I click my IPython notebook, it will open, and here we'll see the full notebook with the pyMETAflow, right? We have some markdown text with some introduction, and for each code, cell code, we have some introduction. So Control Enter to run, or we can push this button here for running. This is the library, the essential libraries. It shouldn't take that long, but I have some other softwares running, so I might be busy. Sometimes I push my computer too far. Okay. Um, so in here I set the directory path, and that will collect the path that we are in. So you don't need to type your path, right? That's the path that I am, and that's the data that I'm importing. So we have a data of 65,000 points and 26 samples, actually 25, but the first column is the chemical shift column, okay? And we read the metadata as well. That will be the 25 samples, right? 25 columns. So when I run these, this is for creating plots. Sorry, let me make it smaller. So once I run these, it will create another subdirectory, all images, and it will add the spectral image there. Remember that I told you guys that the images are saved as HTML. So you can go and check your data, create zooms, and check for chemical shifts, and you can actually see the some information here. That's the file name. You can add or remove some. Let's just show you. Let's make it easier to check something. Sometimes you have a standard in there as well. So you you want to leave just the standard, or you can compare. You want to compare just some specific data sets. Okay. Anyway, you can do that. Uh, and then you can filter for some regions, right? In here, I'm cutting the ends of the spectra. So I define a starting limit and an end limit as minus 0.5 and 10, just to filter the spectra of everybody as not to see that distortion at the ends, right? Sometimes we don't need to see that. So, but I'm keeping the DSS peak here just because I want to reference again. This is pretty much well referenced, but let's make sure. So I can mask certain regions just substituting by zeros some peaks that we don't want to see. You see, if I go here, this is a solvent peak. This is actually water. I don't want to see that. I don't want that in my statistical analysis, right? So I can remove it. So I set a region as in ppms, and I can run that. It will let you know that that region was removed, and I can remove specific file names as well. But in that case, in this case here, I don't have anything to remove. So anyway, you can just add a file name here, right? Uh, so this is the reference part. That is as simple as putting the DSS as it should be. You can do reference on MNova, but well, I find it useful to do it here, or you can do it both. and it will create you another plot. Now without the regions that you don't wanna see, you can compare with that, right? That is easier to look, right? And the peak, the DSS peak is right in the zero. Okay, we don't need to see that right now again. And that is saying my X limit is between in a window of 0.4 ppms around zero, and it will find any peak that is in that position, and it will place it on 0.0. Okay, and then I can cut the DSS peak out because it's not part of my statistics. Okay, so for the alignment, I can do ICO shift to make it. This is just warning. We don't need to see that. Raft and path. I'm running just by clicking Control Enter. Okay, it will create an image for each alignment, and then we can compare the four spectra, the original one, that will be the original, the alignment by ICO shift, the aligned by path, and the aligned by raft. Then we can decide which one is the best one. Okay, that might take a while. Raft is done, ICO shift is done, path is done. I want to see my comparison multiplot here, that file here. Okay, it will open as a HTML, and you can zoom and check for regions. See, you can zoom and check for regions across your data. Let's get some example here. That might be a good example. Everything's very well aligned because this is good data, right? Okay, so this is aligned by raft, this is aligned by path, this is aligned by ICO shift. And you can walk first region. That's the region that we remove it. Well, we don't want to decide anything right now, but in a series analysis, you will check everything, right? So this is a good example that you need to check for the alignment, right? Okay. So next step, you check it, your aligning method, aligning method, and you choose one. So you decide which one you choose by clicking here. You can copy and click here, and it will create an aligned DF. In this case, I chose path, right? Uh, well, as I told you, you can just export your data for the meta-analysis format and move forward to meta-analysis, or you can do the basic stuff here. Okay, You will create another directory data, and you will place the metaboanalysis there. Okay, the data there, and that's the format. You can read that directly on the metaboanalysis. So for quality control, we filter for total pool just because in the metadata they call the QC pool sample as total pool, and we can create a figure for plotting everything that is total pool as a spectra, just a check, and we want to check if everything is really overlapping. And that's, well, impressive, right? That's cool. And you can select some regions of peaks of interest and check for their standard deviation of the area under the curve, right? So you have figures like that being created and save it as uh, PNG. Okay, well, standard deviation is 0.01. It's quite good. Okay, the Stoxy, that's a good analysis. And I'm just select the driver here, and then I get the image. The image is also save it as a PDF, just because I like it, PDF image. I will update these to get a better HTML figure. That's giving me a hard time right now. That's just warning. And this is another variation of Stoxy where you can choose the model to be used for the online for the Stoxy. We can use linear, exponential, sinusoidal, sigmoid, Gaussian, FFT, polynomial, piecewise, and skewed Gauss. That is just variations that I was exploring for this model fitting. So for now, we can move on with the data transformation, and we can remove the user pooled samples. So I'm just targeting my study samples, well, not mine, the one that I downloaded, and that would be non-smoker versus smoker. So I can filter them, and now I have only them as data. I can do some data transformation, log, square roots or cubic root. If I want to apply in anything, but you can choose which one you use by clicking here and checking your data distribution. It will create a figure, an image for you to compare. That might take a while. Okay, that's run. We can see a distribution for before the transformation and after the transformation. That would be none. Okay. Um, I think that's good enough for now. We can do normalization using those methods. That's done. Then we choose which normalization method we choose to be the best just by clicking there, and we wait for the plot. Let's just go ahead and calculate the scaling in the plot here, just because this takes a while. Actually, to print a plot. Okay. Okay, the plot is done. and we can see the normal distribution of the curve. I won't go into details for that just because I'm just demonstrating the thing, but I would investigate all the different methods and combinations just to check the final result. That is selecting range as a scaling method. That's just warning. That's not error. But that looks normal enough, right? And then we can print like a um, overview of everything that we've done right now. Remove it unwanted regions, and then remove inner regions, and then samples was removed. No sample was removed actually. The alignment method, path, the transformation method, none, the normalization method, PQN, and the scale method, range. And we can uh, create an image for the scale spectra. We don't need to see that right now. And then we can, well, just export that in the metaboanalysis format as well. That will go for that same directory data just to save the data. And then we move ahead for the multivariate analysis. This is doing PCA, and the plot will be saved as a HTML again in the images folder. That's there. Explained variance and plot score. We can open those just to check. Explained variance 98 for the first one, but that's also in the axis. And color coding is based on the classification column from the metadata. That would be the attribute group in the metadata. Again, the classification is not used for the PCA calculations, right? And you can hover over the dots to check which is which. And we can create a loading plot as well, just to check, and another, just save it on there, on the same thing. We can open that, just check. So you can zoom, you can check for details here, you can check for chemical shifts. That's a good thing to do because you can select specific chemical shifts to do Stoxy later, just because Stoxy helps you with the annotations. And that would be a plot for the loadings with the spectrum on the background. But I think this is not very good. Let me try some other thing. We'll update these. So that's a better view. Just for you to go for samples. Samples are in gray, and the loading plot is in blue, right? It's a good view that helps a lot with the interpretation of the data, right? Okay. So same thing for the PLS-DA. We create the PLS-DA score plot, right? That's useful. It's a nice looking plot. The validation. It will calculate again, just to calculate to bring the image for the Q square and R square. And we can create the loading plot for the PLS-DA. The same way we create loading plot for the PCA. I'll just move on here, check it, because the next step would be the calculation of the VIP score, the variable for the VIPs, right? Very important. People, no, the variable important to projection. Yeah, that's taking a while. I have other programs running on the background. Even MC mine, I should have that closed it before. Sorry for that. Okay, that's run. So we have the Q square and R square. Just to check. Okay, we have the loading plot. Save it. Component 1, and we have the that's the VIP, right? It's running. I won't run that just because that's a variation of, well, let it run. It shouldn't take that much. So that's done. That's the table for the VIPs, right? This is save it on here, score plot. Okay, that's not a good plot. I have to make it better, right? And here we can do data and that, just to make sure it will go save for the save on the data folder. Okay, that should be that one. Okay. That's just another plot. We can do the same for orthogonal PLS-DA. I won't run that just to not to waste your time, but that will make you an OPLS-DA plot, score plot, the loading plot, and the VIP scores. We have also HCA, hierarchical cluster analysis, and it will save the dendrogram here. We can open that and check it. Okay, that looks nice, right? Okay, maybe I'll change that to make into the, or maybe color code according to the metadata in the future. Okay, we can also select some regions and do integration for everything, and then to create a box plot. So it keeps printing things. Okay, and I'll save it on inside on another. I'll fix that later, just because that's the same here. Okay, or maybe we can do that. Let's check it. It should save the overlay in here. I'm gonna double check that. Anyway, that's the overlay regions selected. Okay, so it will calculate area under the curve, and then we can do box plot. Okay, for each region, for the different groups, we have a box plot here just to check how it goes. That's interesting for something like a univariate analysis. In the near future, I'll be adding um, SVM and random forest. Again, you can export that to MetaBond list. And now for the data fusion thing, we'll make use of the data metadata file just to check that we have only NMR and bioactivity to be added. If our metadata had the MS file, it would push for NMR bioactivity and LCMS file, right, to be fused and correlated. We can run these. That will put everything in order. Or then we apply Stoxy, calling the driver from the bioactivity, right? So we can create a plot. Remember that my bioactivity data is a mock data, so it doesn't have any value. But we can do the Stoxy calling using the bioactivity as a driver. So what I'm doing is basically asking for the bioactivity, who are the NMR features that are related with that. And that's very helpful, specific for natural products. For some metabolomic study, you can use the uh, what do you call it? Well, your classification analysis, that would be the phenotypical analysis, and create a vector for that, and then you calculate the correlations to get the NMR profile of the highly correlated peaks, right? That's the image produced, and that's it. Okay, thank you, and I hope this is useful for more people besides me. Thank you.